Transposons in bacteria. Transposons are segments of DNA that can move around to different positions in the genome of a single cell. It was first discovered in 1948 by Barbara McClintock in maize. It comprises 40% of human chromosomal DNA. They are also called mobile DNA elements or transposable elements. Transposition, the process by which the sequences are copied and inserted into a new site in the genome. Transposons can be a major source of mutations in the genome. Transposable elements were first identified at the molecular level in the form of spontaneous insertions in the bacterial operons. Such an insertion prevents transcription or translation of the gene in which it is inserted. The antibiotic resistant transposons in bacteria were originally designated by abbreviation T and followed by number 5, which distinguishes the different elements. By arrangement, all newly discovered bacterial transposons are to be designated in this way, even if no recognizable gene is present. Then types of transposable elements, IS elements, transposons, bacteriophage MU. The transposon first discovered did not contain any known host genes, and for historical reasons, they were called insertion sequences, or IS elements are designated IS1, IS2, and so forth. Insertion sequence, genes for transposition, inverted repeats. Then IS elements, the first transposable element that were discovered in bacteria in 1967 were the IS elements. They are the simplest type of transposable elements found in bacteria. It encodes transpose SG for mobilization and insertion. The IS are shorter and do not code for proteins. Ends are all known. Ends of all known IS elements show inverted terminal repeats or ITRs. Then integration of an IS element may disrupt coding sequence or regulatory regions, alter expressions of nearby genes, cause deletion and inversion in adjacent DNA, result in crossing over. So IS elements, inverted repeat, protein coding region, target site directed repeat. General structure of bacteria in IS elements. Then transposition of insertion sequence elements. Original copy remains in place. New copy inserts randomly. IS elements uses host replication enzymes for replication. Transposition requires transposase enzyme coded by the IS element. Transposition initiates when transposase recognizes inverted terminal repeats, ITRs. Target site is the site for integration. Staggered cuts are made in DNA target site. IS element inserts, DNA polymerase and ligase fill the gaps. Small direct repeats about 5 BP flanking the target site are created. Donor DNA, target DNA, transposase make blunt-ended cuts in donor DNA and staggered cuts in target DNA. Then unpaired bases, transposase ligase to single-stranded ends of target DNA. Cellular DNA polymerase extends 3' cut ends with and ligase joins extended 3' ends to IS10 5 ends. Ion base pair target site direct repeats. Then transposons TN. Similar to IS elements but are more complex structurally and carry additional genes. Two types: composite transposons, non-composite transposons. Composite transposons. A composite transposon is flanked by two separate IS elements which may or may not be exact replicas. Instead of each IS element moving separately, the entire length of DNA spanning from one IS element to the other is transported as one complete unit. Then carry genes might be a single or antibiotic resistance flanked in both sides by IS elements. TN1 is a composite transposon. TN1 is 9.3 kb and includes 6.5 kb of central DNA, includes a DNA gene for tetracycline resistance and 1.5 kb inverted IS elements. Transposon, chloramphenicol resistance gene 5 BP direct repeat. Then transposon, inverted repeats of IS element, tetracycline resistance gene, inverted repeats of IS element, inverted IS elements. Non-composite transposons. Lack flanking insertion sequences contain terminal inverted repeats, carry genes. Example might be a gene for antibiotic resistance but do not terminate with IS element. TN3 is a non-composite transposon. TN3 is a 5 kb with 38 base pair ITRs and contain 3 genes in its central region. Then BLA beta lactamase breaks ampicillin, TNPA transposase for insertion, TNPB resolvase for recombination events. Transposon TN3. left inverted repeat, transposase, resolvase, beta-lactamase, right inverted repeat, mRNA. Then bacteriophage MU. It is the longest transposon known so far. Bacteriophage MU carries numerous genes for viral head and tail formation. Temperate bacteriophage MU mutator can cause mutation when it transposes. Its structure include a 35 kb linear DNA in the phage particle that has central phage DNA and unequal lengths of host DNA at the ends. The DNA G segments can invert and it is found in both the orientation and viral DNA. So following infection, MU integrates into the host chromosome by non-replicative transposition. An integration produces prophage DNA flanked by 5 BP target site direct repeats. Flanking DNA from the previous host is lost during integration. The MU prophage now replicates only when the E. coli chromosomes replicate due to a phage encoded repressor that prevents most MU gene expression. MU prophage stays integrated during the lytic cycle and replication of MU's genome is by replicative transposition. MU causes insertion, deletion, inversion and translocation. Structure of MU phage: capsid coating, dsDNA, tail sheath, N protein, base plate, tail fibers and tail tip. Transposase binding site: regulation of transposition, lysis, head and tail genes, head range. Then transposase binding site, ATTR, host DNA, genome organization of MU phage.